Okay guys, so our last chapter in this unit is going to be chapter 3 over cell biology. So after this we will have our first exam covering chapters 1, 2, and 3. Starting off, when did we first find out about the cell? This really came from two individuals, one of which you've probably heard of in like your high school biology class, another one you probably have not heard of. So Robert Hooke is one that's very well known. He basically was a scientist that was studying things, and he created one of the first powerful enough microscopes to look at these really small details that we can't see with the naked eye. He's most famously known for looking at a piece of cork that was sliced really thin, and he noticed that was made up of these great many little boxes, which he called cellulae, eventually coining the word cell. Now, someone that you probably have not heard of is Antoine van Leeuwenhoek. This guy was totally unrelated, but about the same time as Robert Hooke, he also created one of the first microscopes. Again, had nothing to do with Robert Hooke. He created his own version. This guy wasn't even actually a scientist. He was a tailor, which means he made clothing, things like that. So he made a microscope to look at his clothing a little more closely, and he started to see what he called little animalcules, or tiny animals, under his version of a microscope. And these were actually the first microorganisms. So Robert Hooke first discovered the cell. Leeuwenhoek discovered microorganisms. Part of the reason why you don't hear about Leeuwenhoek as often, besides the fact that he wasn't a scientist, was also because he was kind of weird. Once he discovered these, he kind of dropped tailoring and basically started looking at everything he could under the microscope out of curiosity. But it's what he looked at that kind of made people a little standoffish. Started with things like pond water and leaves, things like that, but eventually graduated to teeth scrapings under his fingernails, feces, both animal and human, not just his own. So people tend to give him a wide berth. So this is showing some of those first microscopes. We have come a long way in terms of our technology and what we're able to use, and also we've able now to see even smaller than what these guys first saw. So eventually all of these observations and then a couple other scientists added into what eventually became known as the cell theory. So Schleiden, Schwann, and Virchow were all German scientists that added into the cell theory and kind of put all of the information that previous individuals had gathered, including Leeuwenhoek and Hooke, and they eventually created this overlapping theory of science. Now, one thing to note about theories in the scientific world, a scientific theory is not a guess. When we say a theory in our everyday language, we mean, oh, I have a theory about this. It means you have a guess, you're, making a speculation. A scientific theory is not a guess. It is something that we have tested over and over and over again. It has never been proven false. So we are 99.99999% sure that it's true. Now, there could always be some, new technology that comes about that proves us wrong, but this is kind of the most laid out universal theory there is. So there are three parts of cell theory that all kind of go together. The first states that cells are the basic unit of life. They are the smallest living thing. It's the one single-celled organism that eventually gave way to all of these more complex organisms, including humans. Second part of cell theory states that all living things are composed of one or more cells. Everything that you think of as a living thing is made of cells. Some organisms are single-celled, some are made up of billions of cells, like we are. And then the third one is all cells come from pre-existing cells. This is very important because at the time they had this idea known as spontaneous generation theory. That was very popular at the time, but it basically stated that small things just kind of appeared in existence. And not just microbes, even things like frogs and mice and maggots, bugs, they would see these things they would see frogs coming out of ponds, and they didn't have the, background information to know that they came from tadpoles that had been eggs laid in the water by previous frogs. So they just saw these organisms coming out of ponds. They're like, oh my god, this pond just birthed a frog. Cool. Same thing with maggots. They would see maggots on rotting meat and things, and be like, ooh, this rotting meat makes maggots. We now know that's not true. But at the time, that was the information they had. So eventually, this gave way to spontaneous generation theory. Disproved spontaneous generation theory and said that all cells come from pre-existing cells. So there are several experiments that kind of led into this disproving of spontaneous generation. Two of the more well-known ones involve Francisco Redi, and then Louis Pasteur was kind of the nail in the coffin for spontaneous generation. But it started with Francisco Redi, that he basically tried to prove that, no, look, these small things, these small maggots on rotting meat come from flies. And so he basically took 2 jars to start with. One he left open so flies could get in and lay eggs. One he covered with like a cheesecloth on the top so the flies couldn't get in. The problem was the cheesecloth has big holes in it, so flies were still laying eggs. They were falling through. Both times maggots ended up on the meat. Finally, he took a sealed jar, so there was no way the flies could get in, no way the eggs could get in, and there was no maggots that came on that meat. Now, there were still some skeptics with this, though. They were like, well, it's because you cut off the air. Things can't live without air. So finally, we had Louis Pasteur did a final experiment with a swan-necked flask. Basically, what he did is he took 3 types of broths in these different types of jars. First one he just left open on the far left there, eventually became contaminated with bacteria. Another one he plugged with cotton up at the top. That one, because nothing could get in, after he boiled the broth to clear everything out, it was still sterile even weeks later. And then to finally prove that the air had really nothing to do with it, he took this swan neck flask that he left open for months and months and months and months and months, and proved that there was no bacterial growth in there. And to take it one step further, in the end, he actually hacked off the end of the swan neck flask, and then within a day, bacteria had started growing in it. Okay, so now we have just a general background on cells. Let's look at the actual parts and organelles inside a cell. So starting with probably the most important organelle. If you don't know, organelle just means like a little organ. So our cells have these little things inside them called organelles. Each has a specialized part. Probably most important is the nucleus. So there are several distinct parts of the nucleus we will look at, starting with the nucleolus, then the nuclear envelope, and finally the DNA itself. So the nucleus overall is one of the largest cytoplasmic organelles. Its main function is to contain all of our DNA. All of the genetic information of an organism is stored here. Now, the amount of the DNA depends on what organism you're looking at. Humans, for example, have 46 chromosomes, and then different organisms have fewer or more than that. I think the most we've seen is like 115 chromosomes, all the way down to just 1 chromosome. Some other terms that you should probably know is that a gene is a little section on a specific chromosome that codes for 1 protein. So this is a simplified version, but say on chromosome, 14 on the human chromosomes, say on chromosome 14, 1 section codes for hair color. That is 1 gene, the gene for hair color. We also need to know about chromatin. So chromatin is just the DNA in the nucleus, but when it's called chromatin, it just means that it's loose, just kind of hanging out in the nucleus. It becomes known as chromosomes when it becomes really tightly coiled, and that's when it forms those kind of X shapes that maybe you've seen before. If not, you will see here shortly. So chromosomes and chromatin, they're the same thing. They're the same material of DNA. It's just chromatin is when it's loose, chromosomes is when it's compacted really, really tight. So one of the specific parts of the nucleus is known as the nucleolus. This almost looks like a nucleus inside the nucleus, that little center area there. This guy's important because this is where we're going to make RNA, specifically ribosomal RNA or rRNA. It's going to be the site where we produce these, and eventually we're going to ship those out of the nucleus into the cytoplasm. From there, ribosome's job is going to be to synthesize or to make proteins, but this is where the ribosomes themselves get made. We also have the nuclear envelope. So this is basically a second cell membrane, specifically around the nucleus. So it's another double membrane, just like we have around our cells. We have a phospholipid bilayer. This actually has 2 of them, as you can see in the image there. 2 layers of that phospholipid bilayer, and this one's also going to have pores in it to allow things in and out. So this is going to regulate not only what comes in and out of the cell, but what comes in and out of the nucleus. So just because something gets into a cell doesn't mean it automatically has access to the DNA. It's kind of like if you get into a club. You have to go past the bouncer just to get into the club, but that doesn't mean you automatically walk to the VIP section. You have to get past yet another bouncer to get to that section. Okay, moving on into the cytoplasm and the cytosol. So cytoplasm essentially is all of the material inside the cell and around the nucleus, all of the stuff, not just like the fluid stuff, but also all of the other organelles that we'll look at except for the nucleus. Now, in high school bio, you probably knew cytoplasm as just like the fluid inside the cell. That's kind of true. Specifically, cytosol is the fluid-like stuff, the jelly-like stuff in the cell, but cytoplasm is the cytosol plus all of the organelles that are suspended in the cytosol. This is also where we'll see at the very end here, just for like a hot minute, we'll look at cell respiration. This is where the first step of cell respiration, known as glycolysis, is going to happen in the cytoplasm, specifically in the cytosol. Next, we have the endoplasmic reticulum. I am 100% fine if you just call this the ER because ain't nobody got time to say endoplasmic reticulum over and over and over again. So I'm fine with just ER, whether it's on exams, lab practicals, whatever it is. So this is basically a network of membranes that are going to kind of fold in on each other, and we're going to have a lot of chemical reactions happening here. Specifically, we are going to have 2 distinct parts with 2 different reactions happening. We're going to have what's known as the rough ER and the smooth ER. The rough ER is going to be rough. I know, shocker. You can see in the image there, it almost looks like it has little studs on it. We'll talk about those in a second. And then the smooth ER is the one that doesn't have any of the studs on it. So starting with the rough ER, this is called the rough ER because it's studded with ribosomes. Remember those guys we made in the nucleolus and then sent out in the cytoplasm? Some of those will stay free floating in the cytoplasm, but some of them will attach to the rough ER. Now remember we said ribosomes' main function is to make proteins. So the rough ER is going to deal with the proteins that those ribosomes are making. They're going to finish off the proteins that were made. So they're basically going to kind of like repackage and get those ready after the ribosomes are done with them. Then the smooth ER is called smooth because it doesn't have any ribosomes on it, so it's physically smooth. So this time, instead of dealing with proteins, the smooth ER deals with lipids. Lipids is just a fancy term for fats, remember? So this guy is going to make lipids or metabolize lipids. It's also going to make some cholesterol, which is a form of lipid. It's also going to make some hormones, which, remember, are a type of lipid. So I remember this one because the ribosomes, remember, job is to make proteins, so the rough ER deals with proteins. I remember the smooth ER deals with lipids or fats because most fats are smooth. Think about like, butter, cream cheese, meat, smooth, right? All smooth. So here's just another look at that ER. You have the rough on the left-hand side there with those ribosomes, and then the smooth on the right. Next, we have the Golgi apparatus. So this is going to be stacked kind of flattened sacks, kind of folding again back in on themselves. This guy's function is kind of acting like the FedEx or like the Amazon of the cell. It's going to modify, package, and then transport all of the things that were made in the ER, all of those proteins and all of those lipids. It's going to do this by putting those products in transport vesicles, and these are going to kind of bud off of the edges of the Golgi apparatus that we'll see here in a second. From there, they'll either be sent either outside of the cell through a process known as exocytosis, or they'll just be sent somewhere else in the cell. So this is a nice image showing that Golgi complex or the Golgi apparatus or the Golgi body, whatever you want to call it, but you can see it folding in on itself, and then the edges of those folds kind of budding off, popping off. Those are the transport vesicles that are filled with the finished and transport ready lipids and proteins. Next, we have mitochondria. If you remember nothing from your high school bio class, you probably remember mitochondria as the powerhouse of the cell. That is true. Mitochondria's job is to make a ton of ATP, which remember is our energy molecule, specifically in a process known as aerobic respiration that we will briefly talk about a little bit later. But basically, it's going to make a ton of energy. Now there are two main parts of the mitochondria I want you to know, the matrix and the cristae. The matrix is going to be the cytoplasm of the mitochondria. It's going to be the inside jelly-like stuff. And then the cristae are going to be all the inner folds that you see in the images there. These are important because that's where that aerobic respiration is going to happen. It's kind of like, if you think about fancy kitchens, the nicer the kitchen, generally the more counter space you have. That's basically what these do. They create more counter space, quote unquote, inside the mitochondria, because the more space they have, the more surface area they have, the more of this aerobic respiration they can do at once, which means the more ATP that they can pump out for the cell. Next, ribosomes. We already mentioned that these guys are used for making proteins. Remember, there are a couple different locations that we'll have for these. Originally, they're found in the nucleolus. They'll eventually move into the cytoplasms, and from there, they'll either stay free-floating in the cytosol or they'll attach to the rough ER. Now, there are several types of ribosomes, all of which are used for protein synthesis. But we will see ribosomal RNA, or rRNA. Its job is going to be to actually build the proteins. Transfer RNA, or tRNA. Its job is going to be carrying pieces, the little baby pieces of protein, to the rRNA while it's building, basically like the fetcher, right, for the rRNA. And then finally the messenger RNA, or mRNA. This is going to be like the blueprint that rRNA is reading as it's building the protein. It's like the instruction manual for how to build a protein. Next we have lysozymes. These ones are my favorite. So these are basically little balls of acid, or little buckets of hatred inside the cell that are full of digestive enzymes and acids. So basically their job is going to be to break down anything that the cell doesn't want. So anything that it wants to get rid of, it's going to shove into a lysosome, and then the acid and the digestive enzymes will tear it up and spit out the parts. There are lots of examples of these. I don't really care you know these two specific examples. But for example, our liver has tons of lysozymes that's only function is to break down a product known as glycogen. Glycogen is one of our big energy storage molecules that when we break it down, it will release a ton of glucose, which is our main sugar molecule we use for energy. We also have white blood cells that go through phagocytosis, or phagocytized bacteria, basically where they kind of engulf and eat a smaller bacterial cell that's not supposed to be in our body. They'll also do this to injured or worn out cells, or parts of cells that have lysed, chew up the parts and spit them out. So this is a nice image overall of several of the parts we've talked about. You see the endoplasmic reticulum at the top there, specifically the rough ER with the little ribosomes on it. You see the Golgi apparatus with the little vesicles budding off of it. And then you also see some lysozymes in there. Lysozymes, remember, we said are little buckets of acid. You can see one merging with a food vacuole, a little ball of food inside the cell. Lysosome binds with it, releases the acid, the digestive juices. It's going to break down the food into parts and then send those either into the cell or out of the cell through exocytosis. Next we have peroxisomes. These are again just little sacs inside the cell. As far as identification goes, they look exactly the same as lysosomes and vacuoles and vesicles. They're just little circular things inside the cell. So as far as that goes, I don't care. You can identify those too much differently. We'll talk about that in lab. But the function you need to know is different. Its job is to detoxify things. So for example, like alcohols, toxins, hydrogen peroxide, things that if it builds up in the cell, it becomes toxic and can hurt the cell. So they take them and turn them into products that won't hurt the cell. And we have a ton of these in different areas of our body. Two of them, the highest number locations are the liver and the kidneys. Next we have vesicles and vacuoles. They're basically the same thing. They're basically just little sacs that are used for storage or movement of different products. So for example, here we see again the rough ER and the Golgi apparatus working together, and you can see the vesicles budding off the Golgi apparatus and teal there. They're just moving those products around. Essentially the same thing. Vesicles tend to be the ones moving things. A vacuole tends to be more like a storage unit that's stationary, but basically the same thing. Moving on to the cytoskeleton. So cyto means cell. Skeleton, of course, means skeleton. So this is going to be the skeleton of the cell, or the skeletal structure of the cell. So its main job is going to be support, and then also generating movement inside the cell, which is going to be kind of odd. So this is going to be made up of little proteins that are shaped in the form of rods or tubes known as microfilaments and microtubules. So anytime you generally see an image of cell and cell parts, or really even the models you'll see in lab, usually the cytoskeleton is pulled out and it's not shown, or you just see like a piece of it here and there. But really, like you see in the image here, the cell is full of this cytoskeleton. It almost looks like scaffolding going everywhere, or pipes going everywhere. That's because really nothing is free floating inside the cell. There's very little that's freely able to move. Almost everything is attached somewhere. Just like inside our body, there's really no free floating stuff. Everything is attached to something else. So this is just showing some of the different types of those proteins. We have microfilaments and microtubules. We said microfilaments are the teeny tiniest. Microtubules are these really big guys. Technically, there's an intermediate filament that's kind of this middle ground, but that doesn't matter too much. So now looking at a little bit closer at these. So microfilaments, we said these are kind of the real thin thread-like guys. These will be especially important to us when we get to the last unit in A&P1 when we look at muscle contractions. We'll also see them a little bit in the next unit when we look at the skin. So we will have myofilaments, myo meaning muscle, but these filaments that are known as actin, they will work for muscle contractions, and then keratin is going to work inside the skin in the next unit and kind of hold the skin together. And then finally, microtubules. So these are going to be organized more in like bundles rather than threads. These have several jobs. One of the biggest is just anchoring everything down to the cytoskeleton inside the cell, but also this is what's going to make any kind of movement inside the cell. So including things like making flagella and cilia that we'll look at, like flagella in sperm cells you've maybe seen allows them to swim, and then cilia we'll see like in A&P2 when we look at the respiratory unit and the digestive unit. They're these little hair-like projections that basically beat back and forth, back and forth, back and forth, and cause fluid to kind of move past the cells. Another reason that we care about these microtubules is we will see them in mitosis later in this chapter. They will form these things called spindle fibers that will yank the chromosomes around during cell division. Speaking of cell division, we can't forget centrioles. These are little barrel-shaped organelles. I don't care that you know that they sit at right angles, triplets of 9. Nobody cares. I do want you to know that their main job is to help with spindle formation or mitotic spindle formation. Basically, if you see in the bottom image there, they're the little red guys on either end. That's what the spindle fibers, those microtubules, are going to attach to, and then also attach to the chromosomes. So this is what's going to kind of give an anchoring point for those microtubules to yank those chromosomes around for cell division. And then last but certainly not least, probably the most important other than the nucleus organelle we have is the plasma membrane, essentially the skin of the cell. Now there is a lot going on in the plasma membrane. One of the biggest things we'll see is the phospholipid bilayer, those little yellow guys that we'll talk about their structure here in a minute. We saw them a little bit in the chemistry chapter. But these are going to create the main force of keeping things in or out of the cell, because we'll see the middle layer of those phospholipids, the little zigzaggy lines known as the tails, are going to be hydrophobic. They don't like water, so it's going to create this non-polar region that most things can't pass through. Versus the little round heads, those are polar or hydrophilic, so they like water and they deal with water, so they create kind of this impassable layer for most things in the cell when you put these guys together like this. Three other important pieces that we'll look at a little bit more are cholesterol, the little blue guys stuck in the phospholipid bilayer. Cholesterol, remember, is a type of lipid. So these are basically kind of going to act like studs in a drywall wall. This is basically going to give like support and structure to the phospholipid bilayer. We'll also have carb chains or carbohydrate chains. They're in green. They kind of look like little tree branches coming off the top of that. Some of them are called carbohydrate chains. Some of them are called glycoproteins when they're attached to a purple protein there. Some are called glycolipids when one's attached to a lipid. Basically, just the little green things are carbohydrates. These guys have a bunch of functions. One of the biggest ones we'll see is it gives the cell kind of a fingerprint, and we'll look at that here in a second. And then finally we have all the purple guys. Those are different proteins stuck in the cell, and they have lots of different types of proteins. We'll see some are transmembrane proteins, some are channel proteins, some are just anchoring proteins. We'll get to them. So overall, the job of the cell membrane is going to be a barrier. It wants to decide what can come in and out of the cell, and it's going to do that with that phospholipid bilayer. This is going to make it a selectively permeable barrier, which means it picks and chooses what can move. So it is how it regulates what can move into and out of the cell. Another important term to know for plasma membrane is this term fluid mosaic model. That basically states that all the things in the plasma membrane are not nailed in like you have like a structure in a building. They're almost free floating. They kind of move around a little bit, almost like an olive oil consistency. They're able to kind of shift around as needed a little. Now it's not like you have something going from, one end all the way around to the other end of the cell, but they can just shift a little bit in place. And then I just listed what we talked about with the carbs and the proteins and cholesterol in the bottom there. So when we're looking at these proteins, we have different types of proteins we mentioned, starting with integral proteins. These are ones that are going to extend all the way across that phospholipid bilayer. These are the ones that are generally going to act as channels or transporters, sometimes as enzymes or receptors. Some of them will also have a carbohydrate chain attached to them. That's when they're called a glycoprotein. These are ones that are going to mark the cell as self or non-self. These are the ones that are going to basically give your cell a fingerprint saying, yes, I am a Kristen cell, I belong here. This is very important for us because we have an immune system that will attack anything that doesn't say, yes, I belong here. So any bacterial cells, viral cells, whatever it is, even things like blood transfusions, tissue typing, if it doesn't match, if it doesn't say the right, hey, I belong here, our immune system attacks it. And then we also have peripheral proteins. These are ones that are basically going to lie on either side of the cell membrane. Most of them are going to be on the inner surface of the cell membrane, on the inside of the cell. Every once in a while we have a couple on the outside, though, acting usually as receptors. The main job of the ones on the inside is to act as anchors for the cytoskeleton. Okay, so now we're going to take a brief look at how cells communicate with each other. We're going to look at 3 ways that they interact or talk to their neighbors. So first we're going to have tight junctions. So these are basically going to be connection points between cells that create watertight seals. So no fluids can get past the cell line like you see in the top image there. These are really important in several areas in our body, including the blood-brain barrier, which keeps things separated from the brain. We also have it surrounding all of our urinary system and most of our digestive system as well, keeping all the juices in the tubes they're supposed to be in, not just free floating in our body, because that's gross. Next we have desmosomes. These are basically going to act like Velcro. They're similar to tight junctions in the fact that they hold cells together, but these aren't waterproof. These are more just kind of keeping things from ripping apart. We find these mostly in our skin, but a couple other places as well, like heart muscles and things like that. But mostly in the skin. For example, you can move your skin around, like if you pinch your skin or something, it moves with you to a point, right? But it's not like as soon as you move it, your finger like goes through the skin, right? If it does, you're pushing too hard and you need to call somebody. And then last we have gap junctions. These are going to be actual communication tools for neighboring cells. These are like little tunnels, like you see the little pores in the image there, where they're going to be able to pass ions and essentially like pass notes back and forth to the cells, allowing them to communicate. Again, we have lots of places in the body that use these. One of the most important probably are in heart muscle cells. In the heart, they're known as intercalated discs, and we'll see these in the end of the semester when we look at muscle cells as well as again in A&P2. But intercalated discs allow all of the cells of the heart to work together at once so the heart pumps as one organ. Otherwise you'd have parts of the heart pumping, some not pumping, and you wouldn't be able to move blood and you'd die. So here's just showing all 3 of those together again. So we have the tight junctions on the left, the desmosomes acting more like Velcro, and then the gap junction little tunnels for communication. All right, now let's look at cell transport, how we get things into and out of the cell. So first off, there's a difference when things come in or out of the cell. We'll look at how we actually move things here in a second. But when we're moving something into the cell, it's known as endocytosis. When we move something out of the cell, it's known as exocytosis. So endo meaning inside, exo meaning exiting or outside. Now, there are two types of exocytosis. I don't really care that you know the difference, but just kind of showing this here. Technically, you have constitutive secretion or regulated secretion. Essentially, the difference here is only that in regulated secretion, there's little receptors that you see in green. They kind of look like little green Ys on the plasma membrane, that when a molecule binds to them, that will signal the cell membrane to kind of pinch in and form a little vesicle around the materials. But otherwise, it's exactly the same process. There are also several types of endocytosis. The two big ones that you for sure need to know are phagocytosis and pinocytosis in the green box there. But we also have receptor-mediated endocytosis. It's almost exactly the same procedure. You're just bringing something into the cell. It just varies a little bit. With phagocytosis, it's sometimes known as cell eating because it's bringing in large products. Also, you tend to have the cell membrane kind of reaching out, like you see in the image there, like grabbing the particle and then forming a vesicle around it. Pinocytosis is more known as like cell drinking because this time the cell membrane is going to kind of pinch in and like slurp some of the fluid inside and before it pinches off. And then receptor-mediated endocytosis is almost the exact same thing as pinocytosis, except just like in the second example of exocytosis, you have the little receptors, you have those little, this case, red little Ys on the outside of the cell membrane that when something binds to them, that signals the cell membrane to start kind of pinching in. So now that we've seen how cells can initiate movement, let's look at how molecules can move on their own. So remember, the important thing about the cell is that it has that selectively permeable membrane, which means movement is going to be regulated. Now, with that regulation, some things are going to move easily, others are going to need help moving. The first type of movement we're going to look at is diffusion. Diffusion is going to be the movement of molecules from high to low concentration without the use of ATP, which remember is our big energy molecule, which means we don't have to force anything to happen. Molecules are like people. We naturally want to spread out, we naturally want our own space. If you've ever gone to a party and it's too crowded and you have like, you know, the same girl elbowing you like 14 times as you're like trying to dance or just trying to talk to someone, it gets you agitated, right? And at some point you need to like step out, you need to spread out, step into the kitchen or like on the patio or just leave, right? Molecules are going to want the same thing. They naturally want to spread out, they naturally want their own space. So that's why we don't have to force this. We don't need to use ATP for diffusion because they naturally want to go from high to low concentration. Same thing like when you're in line at Walmart or whatever trying to check out. If you're like 8 people deep, right, in the checkout line and they open another register, nobody has to force me to go to the other register, right? I'm jumping over kids, pushing old ladies out of the way to get there first. Just kidding. I would never push an old lady. I would definitely hop over a kid though. So let's do an example here. Let's say we put these salt molecules or something in this water. What's going to happen given time? They will eventually spread out through diffusion. And they will do this until they are totally separated, until they have as much space as possible, until they're evenly distributed. Now let's add a semi-permeable membrane in there. Now let's say we have the little pink membrane. What will happen given time? In this case, because these molecules can move across the membrane, they will still spread out. As long as they can get through the membrane, they will. We'll see here in a second what happens when they can't freely move across that membrane though. So that's diffusion, high to low concentration with no ATP. Generally the things that can go through diffusion are lipid soluble, which means they can mix with lipids, they are non-polar. These are important because remember that inner layer of the phospholipid bilayer, the little tails of the phospholipids, those are non-polar. So anything that's non-polar can generally go through the cell membrane no problem, especially if it's small. So generally things like oxygen and carbon dioxide, CO2, are freely able to move across the membrane, which is good because your cell constantly needs oxygen and it's constantly making CO2 that if it builds up, it can be harmful. So we want these to be able to freely move. We don't want them waiting on like a shuttle bus to get them out of the cell. Water is also something that can generally freely move. There are some cases where it's regulated a little bit, but for the most part, water can freely move across the cell. But when water goes through diffusion, it's known as osmosis, which is the same thing as diffusion, it's just diffusion of water. So it's still water moving from high to low concentration, no ATP needed, but it's just the diffusion of water. Now, generally, water is a bigger molecule and it's polar. Right? Water, of course, is going to be hydrophilic, water-loving, polar. So it's not going to be able to just shoot through the cell membrane. It's going to have to go through some kind of pore or channel protein. So that's all well and good for smaller molecules and for those non-polar molecules, but most things are polar. Most things are larger and water-soluble molecules, things like sugars, salts, proteins, amino acids, which means they cannot diffuse across the membrane. They can't just jump through the cell membrane like oxygen can. So most of these are going to have to move through protein channels or carriers like water does. In this case, we're going to have two forms of movement: facilitated transport, which is also sometimes called facilitated diffusion, and then active transport. So facilitated diffusion or facilitated transport is still things moving from high to low concentration and spreading out, and there's still no energy required. The only difference between just straight up diffusion is that they require a doorway. They require a protein, like you see in the image there, allowing them to move. Similar to how water moves through osmosis, it has to go through a channel because it's a little too big. Same thing here. This would be just like at the end of lab, when you're done for the day. You wouldn't just bust through my wall to get to your car, right? No, you'll go through the doorway. At least, I hope so, right? Please. Same thing for these. They'll just go through the doorway. Active transport, on the other hand, is where things change up. This one, because it's active, it's going to require energy. It's going to require the use of ATP. And this is because now things are moving against the concentration gradient. So far, for diffusion, for osmosis, for facilitated transport, we have had things moving from high to low concentration, things spreading out so they naturally want to go. Active transport is the opposite. We are having things go from low to high concentration. We are forcing it to go somewhere that's crowded and it doesn't want to go. So we have to put in some energy, some ATP, and make it go. This would be just like if I was the next person in line in my Walmart line and the lady shut down her register and the only other line had 14 people in it. They're going to have to bring over one of the guys in the little security vest to put my ass over there because, no, I've done my time, I'm almost out, right? They're going to have to force me. They're going to have to put in some energy. Same thing with these molecules. Now let's look at tonicity. So tonicity is defined as how the concentrations of solutes in a fluid are going to put pressure on membranes and influence the movement of substances. So we've seen how things can move and how we can force things to move. Now we're going to see why things need to move. So starting off, we have three definitions that we need for tonicity: isotonic, hypertonic, and hypotonic. And these are different levels of solute concentrations. Another term that you should know if you don't is solutes. Solutes just means something that's dissolved in a fluid. So we'll use examples like salt and sugar over and over, but it could be things like proteins, nucleic acids, could be anything dissolved in some kind of fluid. So that's what we mean when we say solutes. So now back to our three definitions. When we have an isotonic environment, it means that two solutions on either side of a membrane are going to have the same concentration of solutes. And we'll see what that causes here in a second, but essentially it's going to be equilibrium. It means things are going to be as spread out as they can get, so really nothing is going to move, or you're going to have equal movement, so it's going to be balanced out. Now, if we have a hypertonic solution, this means that the solution on one side of the membrane, this particular solution is going to have more solute in it than the other. So more solute, less water. Because if you think about it, if you have more stuff in a space, that means there's less room for anything else. So if you have more solute, it means you have to automatically have less water. So a hypertonic solution has more stuff, less water. I tend to remember this like a hyperactive kid has had too much sugar, right? So that's how I remember hypertonic. Hypotonic, on the other hand, this is a solution on one side of the membrane that has a lower concentration of solute than the other side. So this time we have less stuff, less solute, and thereby more water. So this comes into play because we have that semipermeable membrane. We have that cell membrane present. With that, most molecules are not going to be able to freely move whenever they want across the cell membrane. One thing that can usually move, though, is water. Usually, not always, but usually. So when we have substances that are unbalanced, when our solutes aren't balanced like they want to be, when they are not in the isotonic environment, water will move to compensate. The problem is there's only so much space, again, on either side of that membrane. So remember, if there's more solute, that means there has to be less water. So really what we'll be looking at is how much water is in a space, because water will want to move just like the solutes will move. So let's look at an example to kind of make this make sense. So say we have this solution here, set of solutions with that membrane down the middle. To start with, we'll say that salt NaCl can move freely. So in this case, we have side A is a 25% salt solution, side B is a 5% salt solution. For this, where is sodium chloride, where is salt going to want to move? It's going to want to move into side B, right? Because it's going where it's concentrated, where there's more of it, to where there's less of it, right? So if the sodium chloride can move, it will go from side A to side B. But the problem comes in with that semipermeable membrane. Most of the time, salt cannot freely move. So to compensate, water will move instead. So this is where it tends to get tricky because sometimes students have trouble thinking this way. If I say you have a 25% salt solution, that means you automatically have 75% of it as water, right? Because there's only so much space. If 25% of it is salt, the rest of it must be water. Same thing for side B. If I say you have a 5% salt solution, it means you have a 95% water solution. So in this case, if we have side A as 75% water, side B as 95% water, where will water move to change those concentrations to even out? Into side A. So water will move into side A to compensate for that concentration. We can also do this as an example with like a cell in a beaker of water. So in this case, let's say we have a beaker of water that's pure water, no solutes in it, but we have a cell that's 15% sugar, which remember means then it has to be 85% water. In this case, let's say the membrane means nothing. Let's say where sugar will move if it could. It would go outside of the cell, right? It would go into the water. But because we have that cell membrane, most of the time sugar cannot freely move, right? The solute cannot move. So to compensate, we have 100% water outside, the pure water outside. We have 85% water inside the cell, so water will move into the cell. Okay, so these are examples that I could give you on like an exam of where either the solute is moving or where water is moving. So the reason we give a shit about tonicity is because it can change how our cells function. In fact, it can kill our cells pretty easily. So here, I know the picture's a little blurry, but here we have images of cells in isotonic, hypotonic, and hypertonic environments. So the one on the left, the cell is in an isotonic environment, which means the amount of stuff inside and the amount of stuff outside the cell is balanced. In that case, that's the happy place. There's no water moving, there's nothing kind of going wrong, everything's happy. But if we put a cell in a hypotonic environment, remember hypotonic means there's less stuff, more water. So that means there's less stuff, more water outside the cell, and the inside of the cell in this case is hypertonic, with more stuff, less water. In this case, remember the stuff can't freely move because we have the cell membrane, but water can. So to compensate, like you see with the white arrows, water is going to push into the cell. This is a problem for our cells, like you see in the bottom half of that image under hypotonic, because it will cause our cells to swell up and eventually pop, just like you can only fill a balloon so far before it pops. Our cells would do the same thing, which is bad. Now let's say we put one of our cells in a hypertonic environment on the far right there. Remember, hypertonic means there's more stuff, less water. So now there's more stuff outside the cell, less stuff inside the cell, and vice versa for water. Again, the stuff can't move, but water can. So this time, water is going to leave the cell. This is a process known as crenation that's going to cause our cells to shrivel up and die like a little raisin, which is also bad. So basically, anything other than an isotonic environment will kill our cells. All right, and then the last section we're going to look at in this chapter is genes and gene expression, where we'll look at DNA replication and protein synthesis. So starting off with the cell life cycle, we've talked about this in lab before, so hopefully this is a refresher, but we'll talk about it one more time just in case. So remember, the cell goes through a life cycle just like we as humans go through life cycles, right? We go through phases in our life where we go from kids to school age to college to young adults, then we get married, we have kids, and then we're old and wrinkled and whatever. Cells do similar things. They just, of course, aren't the same thing. It's not like a cell goes to college, right? But the cell does go through its own set of life cycle phases. And those 4 phases are G1, S phase, G2, and then mitosis. So the G1 phase, or the growth phase, is just where the cell starts growing. It's just been made, so it needs to grow up a little bit. Then we go into the S phase, or the synthesis phase. This is where we're going to duplicate the genetic material. We're going to make more DNA, getting ready for mitosis. And then we go into G2 phase, which is another growth phase, specifically used to make a bunch of proteins. Now these 3 phases together, G1, S, and G2, are collectively known as interphase. This is where the cell is living its life, doing whatever things that cell does. That's where the cell spends most of its time. About 95% of its time is spent in interphase. The other 5% is spent in mitosis. Now this graph is not sized correctly just so you can read it well. It makes it look like about 1/4 of its time is spent in mitosis. It's not. It's only about 5% of its life is spent in cell division. So interphase, the cell living its life. Then we move into mitosis. It is very important that you realize that when a cell goes into mitosis, all other cell functions and processes stop. Mitosis is a very complicated process where we make sure that we move things around properly and we make brand new cells. And there is nothing worse than trying to do something important and you're distracted by 14 other things. Most of you guys know that you're worried about jobs or kids or whatever else is going on while you're trying to listen to this lecture right now, right? Not as easy as it looks. We don't want our cells to be distracted like that. So we basically shut everything down for them to go through mitosis and then ramp everything back up again. So then once we get into mitosis, we'll have 4 phases, which we'll look at briefly again, starting with prophase into metaphase, anaphase, and then telophase. We also have this process known as cytokinesis, where we kind of mix all the other stuff up and send it into the 2 new cells. And again, we'll look at that here in a second. Another sort of kind of phase that we have is in the blue box at the bottom there, the G0 phase, or the basically like stop phase. This is where some cells, not all cells, but some cells can jump out of this cycle of interphase mitosis, interphase mitosis, interphase mitosis, and basically just take a breather. They can stop. This is usually for the cell in order to get really good at doing one thing. And lots of our cells do this, but the 3 most common are our nerve cells, muscle cells, and our liver cells. Now some cells can go into this G0 phase for a little bit and then jump back into interphase mitosis. But some go into it and they stay in it forever. One example are our nerve cells. Our nerves go into G0 phase and stay there forever, which means we never get new nerve cells because they never go through mitosis again. That's why when you get nerve damage, it's so damning because you don't get any new cells replacing the damaged ones. So the overall job of mitosis is going to be to clone the cell. Take those 46 chromosomes for humans and make 2 brand new cells with the exact same 46 chromosomes. The problem with cloning is it can be dangerous because if there's damage in the original cell, you're going to copy that into the 2 new cells. Just like if you make a copy of like a piece of paper or, copies of your tax returns or whatever it is, you're not changing anything in the copy. You're just making the exact same thing again. So any DNA damage we have will get copied as well. So what are some reasons that mitosis is so important? The biggest one is growth, right? We develop in the womb and grow from a child to an adult. That is a big size difference. I don't know if you realized. So in order to get that growth difference, we of course need to add new cells. In addition, we are constantly bumping and breaking and hurting things, and things just generally wear out. So we're constantly fixing things, broken bones, pimples, torn skin, whatever it is, we need to fix those damaged cells. And then general maintenance as well. Your hair grows out, your skin wears off, you're constantly sloughing off skin, your bones and joints wear out. So we need to maintain those and make replacements for them. Something that's just kind of interesting that you don't necessarily need to know is identical twins. This happens when a zygote goes through mitosis as soon as it gets fertilized, and both original cells actually stick around, but they don't stick together. So that's how you end up getting twins, or like triplets or whatever, same process, just more. But this is why they have the same DNA, because they actually came from the same original cell. Remember, it's also important to know that mitosis is a form of asexual reproduction. This means that our cells do it all by themselves. If you put A in front of a sciencey word, it means no, none, or not. So no sex happening here, which is good. Can you imagine if you needed that every time you wanted to grow your hair out? No thanks, ain't nobody got time for that. So before we can jump into mitosis, we first have to replicate the DNA in that S phase, or in that synthesis phase. Otherwise, one of our new cells would have no DNA, or we'd split up the DNA and not have enough chromosomes, and the cell would die. So this happens, remember, during interphase, in that S phase. So we're not going to get too deep into this, we're just going to overview really quickly of how this happens. But we've talked about the structure of DNA, how it has those four nucleotide bases, adenine, thymine, cytosine, and guanine, and how they match up A to T and C to G. So essentially what happens is we are going to unwind that double-stranded helix, and we're going to read each side of that opened DNA molecule. And as we read it, we're going to build new DNA molecules on top of it. So if you look at the bottom half of the image there, it shows how we have one strand of DNA in blue, we open it up, and then as we read it, we're building the new pieces of DNA in orange there. So you end up with this term known as semi-conservative replication, where we keep half of the original DNA strand in each new DNA, and then half of it is brand new. So here's the overall process of making new DNA. It starts with this enzyme known as helicase. Its job is to unwind the original DNA strand. Step 2 then is we're going to have some proteins bind and kind of stabilize the DNA, holding that open in an area known as the replication fork, which is kind of the Y of the DNA splitting there. Then step 3 is we're going to have another enzyme known as DNA polymerase. It's going to bind to what's known as the leading strand. The leading strand is going to be the strand that goes from 5' to 3' end of that DNA, allowing that DNA polymerase to just kind of run down that DNA strand, following the replication fork, following helicase as it goes down the entire strand of DNA, creating one brand new full molecule continuously of DNA. The issue we come to is on step 4 there with the other side of the DNA. This side is running in the wrong direction. Synthesis always happens in the 5' to 3' direction on DNA. For the other strand, which is known as the lagging strand, this is going in the wrong direction. So when we have a DNA polymerase bind to it, it still has to go 5' to 3', even though it's now running away from the replication fork, like you see the arrows pointing away from it there. This means that it basically is going to latch on where the replication fork is and go a little bit, but then the replication fork is running away from it. It's going down the molecule. So it constantly has to do a little bit, okay, come back, bind again, jump off, go back, bind again, creating these pieces known as Okazaki fragments. Eventually, we'll have those bound together in step 5 there by yet another enzyme known as DNA ligase. It'll take all those fragments that were made on the lagging strand and piece them together, like glue them together, finally creating that second full DNA molecule. So once the DNA has been replicated, once we go through that process, now mitosis can happen. And remember, we have 4 phases: prophase, metaphase, anaphase, and telophase. So remember, interphase is just the cell living its life. doing its thing. When we eventually get to prophase, this is where we're first going to have the chromosomes condense. Under the microscope, I call it chunky DNA. But basically, you can start seeing something happening in that nucleus. Then we go into metaphase. This is where we now have the spindle fibers involved. They've grabbed onto the chromosomes, and it almost looks like they're kind of tied in there with ropes. And it's going to line up those chromosomes right in the middle of the cell. That's how I remember metaphase, in the middle. Then we have anaphase. This is where those spindle fibers are going to shorten, and it's going to break those chromosomes apart, start pulling them towards either end of the cell. And then finally, telophase is where the cell membrane is going to start pinching in at an area known as the cleavage furrow. Yes, that's real, unfortunately, but that little pinch point, and it's going to start making the two new nuclei are going to begin forming, putting the chromosomes in each one. Eventually, it will fully separate. Once it fully separates, that's when we have two brand new cells, and they're going to go back into interphase, going through G1, S phase, and then G2 phase, eventually to go into mitosis themselves, and the cycle will start all over again. So now that we have our brand new cells with our brand new DNA, how are we going to use that information? This is known as gene expression, the process of taking DNA and turning it into proteins. Proteins are what's going to create all of the physical things that you see on an organism. This is known as the central dogma of biology, or like the big main concept of biology. DNA to RNA to proteins, DNA to RNA to proteins, DNA to RNA to proteins. Do you think I said it enough? So overall, human DNA includes all of our chromosomes, which for us is 46 chromosomes that are paired into 23 pairs. On each of these chromosomes, we are going to have genes, little sections of chromosomes that contain the genetic information for making a single type of protein. So this is kind of like your textbook. Our entire genome, all of our DNA is like your entire textbook. A chromosome is like a specific chapter in that textbook. Like this is looking at chapter 3. Then you have a gene is like a specific section or a specific page in that chapter. It's a specific part of the chromosome. So we said that proteins make up pretty much everything we see. It makes up your skin, your hair, your nails. It also makes up things that help us function normally, like hormones. So in order to build these proteins, we first have to know what they're made out of. So remember, the building blocks for proteins, like we saw in chapter 2, are amino acids. There are 20 different amino acids. We're not going to memorize any of them. Ain't nobody care. But you do need to know that there are 20 amino acids, 20 different building blocks that all sort together in different ways to make all these fancy-dancy proteins. And remember, this is different from DNA and RNA because those are made up of nucleotides, where we have the A's, the T's, the C's, and the G's in DNA, and then the A's, the U's, the C's, and the G's in RNA. Basically, what we're going to do is we're going to read what's known as a codon, or a group of 3 bases on RNA and DNA, and eventually make, in place of that, one amino acid, one building block to build a protein. So we're going to go from the process of DNA to RNA to protein. So DNA is first going to be read to create RNA, specifically a form of RNA known as messenger RNA or mRNA. That is then going to be used as like a blueprint and then be read to finally create the protein. So this is broken down into 2 steps known as transcription and translation. So this is kind of a nice overall image here because it really kind of gives you a big overview of replication that we looked at, where we're copying DNA into new cells, and then this process of gene expression in blue. So replication is in green there. It's telling you it's in the nucleus. DNA is copied, making 2 new DNA strands. Then in blue, we have gene expression, where we go DNA to RNA to proteins. It tells you the first part of that, DNA to RNA, is transcription. The second half of that, RNA to proteins, is known as translation. It also is showing you a big difference on where this is occurring. Transcription, because it involves DNA and DNA is stored in the nucleus, has to occur in the nucleus. But because ribosomes are what make proteins and ribosomes are out in the cytoplasm, we then have to take that mRNA molecule out of the nucleus and send it into the cytoplasm in order to do translation. So this is a nice overall image as well. So we have our full cell with our nucleus inside. Inside we have the DNA. It goes through the process of transcription, which we don't care about. RNA splicing doesn't matter, but basically it takes the important parts of the DNA that it needs to make a new mRNA molecule inside the nucleus. From there, it has to leave or be exported from the nucleus into the cytoplasm, and that's where the mRNA will undergo translation and eventually create a new protein. So let's look a little bit closer at these two parts of gene expression. So transcription, we said, was DNA to RNA. Really, the only thing that we're going to change here is we're going to convert the 4 nucleotide bases of DNA, A's, T's, C's, and G's, into the 4 nucleotide bases of RNA, A's, C's, G's, and U's instead of a T. So we're going to flip-flop thymine and uracil. Then once that's done, translation will be RNA to proteins, where now we're going to take the nucleotide bases of RNA and convert them into amino acids. And for every 3 bases, every 3 A's, U's, C's, and G's, we're going to put in 1 amino acid. So we do have specific pairing rules when we're going through replication and transcription. In replication, when we're just making new DNA, we always have that pairing rules of A's to T's and C's to G's and vice versa. In DNA, when we're going through transcription into RNA, the only difference is RNA doesn't have a T. So anywhere that there's an A in DNA and it's trying to read the other side, it can't put a T because RNA doesn't have a T. So you would just substitute a U like you see in that top image there, or top line there, for DNA to RNA. So overall, there are 3 kind of steps to transcription: initiation, elongation, and termination. Basically, start, do the thing, and stop. So in initiation, we're now going to have RNA polymerase. Remember, we had DNA polymerase for replication when it made new DNA. Now we have RNA polymerase when we're making RNA. So RNA polymerase is going to bind to the DNA helix, specifically just to one side. And it's going to read along that side and start building the new RNA molecule in elongation. And specifically, remember, this is a type of RNA known as mRNA. And that's shown in red there in the image. And then finally, it'll reach a termination sequence, and it will basically just release the DNA. DNA will wind back up again, and you have your completed mRNA molecule. Here's another image showing that, and it's a little bit closer. It shows you the matching of the A's, the T's, the C's, and the G's. So because we have our specific pairing rules, when we open up this DNA molecule and we're reading it, we know if we see an A, we know that there would have been a T there, but again, it's RNA, so we put a U instead. Then we see a T, we know that's supposed to match with an A G to a C, an A to a U, C to G, so on and so forth, reading down this molecule. So if you have one side of the DNA, you know what the other side should be. Then, once we've completed transcription, again in that nucleus, it's going to leave and go into the cytoplasm for translation. Now, this is where we're going to have those other types of RNA come into play. We have the rRNA, or the ribosomal RNA, already out in the cytoplasm, and those tRNAs, the transfer RNAs, that are going to start bringing in pieces. So next we have translation. Before that can happen, remember, we have to have that mRNA molecule leave the nucleus and get to the ribosomes for protein synthesis in the cytoplasm. Once there, the rRNA and the tRNA are going to work together to read the blueprint of mRNA to build the proteins. And remember, this is going to happen with a 3-base code known as a codon, and that's going to code for a specific amino acid. tRNA is going to accomplish this with what's known as an anticodon. So an anticodon is basically the matching side to the codon on the RNA, where if you have like CCG on the mRNA, the tRNA molecule will have the match to that, GGC. And then you can see here in this image, that tells it that, okay, I need to bring this specific amino acid. In that particular case, it's the GLY in the bottom right corner there is matching up to GGC. So that's how the tRNA molecule ensures that it's bringing the correct amino acid to the mRNA molecule to build the new protein. Now, you don't have to memorize any of these codes. Nobody cares. We do not care. But remember, I do just want you to know that there's 20 amino acids, 20 of those little colored balls there. The other thing that you should note is that there are far more than 20 possible codes. So there's a lot of overlap. There's a lot of codons that code for the same amino acid. For example, if you look at ser, which is serine, in the second column top there, and then also again in about the middle of the last column, you see there are 6 different codes that lead to the same amino acid. This is so if something happens to the DNA, or the process adds in an extra nucleotide, or messes up a nucleotide, hopefully we'll still end up with the same amino acid. Hopefully we'll still end up with the same protein in the long run. So it's kind of a way to kind of get around any accidents. Now, certainly some still happen, but it's just kind of a safety net for this process of gene expression. And so this is showing that transfer RNA, that tRNA molecule, showing the site where we're holding the specific amino acid site, and then at the top there you see that anticodon. This is where it's going to match up to the codon on the mRNA molecule. And if it doesn't match, it knows it doesn't have the right amino acid, and it's going to get kicked off. And so this is just showing this little process. So you have your first little code coming in, it matches. Then we're going to have the rRNA, the ribosome being built, and you see these tRNA molecules bringing the correct amino acids. Now that one didn't match, right? Its codon and anticodon didn't match, so it got kicked off. The second one matches, and now we have 2 amino acids linked together, starting to build our protein chain. And this will continue all the way down that mRNA molecule, with RNA building, tRNAs bringing the new pieces, mRNA acting as the blueprint, so on and so forth. And then finally you have your finished protein, and for some reason, Pac-Man. And so this is another really nice kind of overview image. It goes through the entire process of transcription translation. So again, we have the nucleus in the top left corner there with the DNA in red. It's being read by RNA polymerase to make the mRNA molecule. Eventually, when that's finished, it will have to leave the nucleus, like you see with the black arrow there, and it will go out into the cytoplasm, where it will line up with the rRNA, and it'll have the tRNAs bringing the correct amino acids in the process of translation, reading, and eventually creating the finalized protein. And again, just another summary image comparing all this, just giving you lots of images to kind of go off of. So we have replication on the far left there, DNA making 2 new DNA strands, and then gene expression, transcription, DNA to RNA, translation, RNA to proteins. Now, that's all well and good, but sometimes things do go wrong. In that case, it's typically a mutation, some kind of alteration of that genetic material. And this can be very devastating because it could cause huge medical conditions or even death if it's severe enough. There are lots of different types of mutations. We're not going to get into them too much. It could just be an incorrect base pairing that happens during DNA replication. Could be of extra nitrogen bases added. Could be that you don't have enough. Maybe some are deleted. Maybe some sections are flip-flopped around. Maybe on the same chromosome, maybe moved to an entirely different chromosome altogether. Generally, the larger the change, the worse the mutation, the worse the side effects are going to be. These can be caused by a lot of things. Chemicals, radiation, high temperatures are the most common ones that can cause DNA damage. This is why things like really high fevers, especially for kids, is so dangerous, because it can cause DNA to start breaking down and cause things like brain damage. Radiation, like especially UV radiation, like lots of sunburns and sun exposures throughout your life, can lead to things like skin cancer. Chemicals like nicotine, if you smoke and things like that, use tobacco, it could cause like mouth, throat, and lung cancer. All of these are caused by mutations. Okay, that's officially it. Everything up until this point is fair game for exam 1, so chapters 1, 2, and 3.